way Giardia grows, if you look, it's actually better in a lower magnification to be able to see. It looks sort of like streaks. Something about Giardia has these weird lines in it. Okay? It grows in clusters, like you can see here, on the lower magnification, and then on the higher magnification. Okay? Just because it's pink on this slide does not mean it's pink in all the other boxes. Okay? A lot of you, was it what?
It was purple on mine.
It was purple on yours? See, a lot of you are writing down, oh, well, this one's pink or this one's blue. With protists, you have to be careful because they're stained using whatever stain was convenient. Okay? It's not like gram staining where pink and purple makes a difference. Can you tell that this is a lot bigger than E. coli? Okay? As a whole, this organism does not look like bacteria. Although it's pink, you should be able to look at that and say, okay, that is way too big. That's not even on oil immersion. Okay? This is just on the third. Okay? So Giardia is in little clusters, and then if you zoom out, it has these like strike through like patterns. Okay? Close that so you don't get a headache.
And what domain would that be in?
You tell me.
Protist or eukaryote?
Yeah, eukaryote.
Okay. Cool.
Because it's not pink.
Maybe I slide to 58. What? Ha! Found it. Okay. Trypanosoma. If you look, I think this one, and I know at least in two other boxes, if you look up, it's only on the far side of the circle. Okay, so this is one of those examples where it's beneficial for you. Hold it up to the light. Find out where the smear is before you put it on the scope. Otherwise, you're just trying to focus glass, which isn't going to help. Okay.
Did you say this one was going to be on purple?
No, this is trypanosoma, not trichomonas.
Oh, sorry.
Okay, when you're focusing, start with the stage all the way down and then work your way up. Oh, yeah. There it is on the first magnification. It's a little blue. You can even see the edge of, so the yellow over there is the edge of the slide. And you can see how close it is to the edge. Okay. Oh, I was like, why can't I see? Okay. Second magnification.
When we review these slides, do we have to like describe the slides, like what we were seeing and stuff like that, or just like domain and
Domain, supergroup, and/or clade. Okay.
This domain is eukaryote.
So all protists are eukaryotes, which means they're all in domain eukarya. I'm not going to lie to you and tell you that I know what clade and supergroup these are on the top of my head, because I don't. trypanosoma. Okay? What's significant about a trypanosoma smear is most of the time it's on the very edge. That's a dead giveaway. Because of the way that you have to smear it, 90% of the time it's going to be on the very edge of your microscope slide. Okay? It's got two distinct portions on a trypanosoma slide. These little bitty tiny things. And then the clusters around it. Again, I'm not going to lie to you and tell you that I know exactly what those structures are. I just know that that's what it looks like. Okay? If you move around, it's very clustered. Okay. There's the edge of the slide. Okay. And then now you're off the slide. Okay. Trypanosoma. That's what we're looking at. Okay. 5. Okay. My euglena doesn't have anything on it. Does anybody have an amoeba? Can y'all look in your slide box and find? Oh no, not euglena. Sorry, pardon me. Yes. My euglena doesn't have anything on it. I can't find one this either. Okay. For euglena. I think the good one of euglena we have pulled already for the exam. That is what you should expect euglena to look like. And actually, euglena has flagella, now that I think about it. So, this is euglena. This is what you should expect to see on the microscope. For some reason, these two euglena have had something crazy happen to them. Okay. What your euglena looks like?
The one back there when it came in.
Yeah. It might be mislabeled. Okay. Euglena. Everybody comfortable with euglena?
Mm-hmm.
It has a flagella. What domain is it in?
Here.
eukarya. Okay. Okay. I'm not going through all of the slides because that would take a ridiculous amount of time. But I can't find the slide that I want. Anybody have 61 paramecium? I'm just gonna steal your slide box. Okay. Alright, I'm staring at two. Okay. Here it is. Oh, no, it's not. On the second, okay? Here's what you're focusing on the first time. Okay? Here's what you get the second time. And here's what you get the third time. That is a little tiny paramecium. Okay? Again, a lot smaller than some of the other protists we've looked at. If you look in your manual, if I went under oil immersion, you could probably see the cilia, but you don't need to do that. So on page I'll get there eventually. 71. You can see the picture of a paramecium. Oh, here's some. So under oil immersion, you would be able to see the cilia, but since we're not under oil immersion, and you're not going to be under oil immersion on the practical, this is what you should expect paramecium to look like. Well, like a dirt. Okay? under the lowest magnification. Okay. Sorry, ideally I would be able to focus it looking at the screen, but the screen is a little delayed. Here it is at the second. And then, good. And third. Okay, that's a little baby diatom. Let's see. Diatoms can be all different shapes. There's your big one. Okay. There's diatoms. What supergroup are they in? Anybody? I think so. Let me double check. Nope. Starts with a C. Mm-hmm. Chromaleobonda is how you say it. Okay. So there are your diatoms.
Do we need to know all of these supergroups?
Yeah, I got you. Oh, there's a lot of stuff.
Okay. Here are some radiolarians. Oh.
What the practicals we should come in for a couple minutes and leave. No.
These are the coolest looking in my opinion, and they're the most obvious. Hint, wink. Okay, here we are under the first magnification.
What is this again?
This is a radiolarian. And what supergroup are these in?
Rhizaria.
Rhizaria. This is only on the first magnification. I'm going to go up one more.
It's under the.
Rhizaria.
Mm-hmm. Here we are under the second. Let me see if I can go to the third.
I'll just write it there.
Okay. I think the second one's better.
Okay. All right. See you later.
There you go.
Which one? Which one? Oh, like the little thing coming out of like a pine.
They look like a bunch of seashells, in my opinion. These are radiolarians. Okay.
I meant to email that you can actually come in this. I figured you can answer me later, whatever.
I'll answer whatever.
Oh.
I don't care if it's like 1 a.m. I'll answer.
I'm going to show you this one and then one more. Okay. Radiolarians. That's what we're looking at. That's what we're dealing with. Where is this? There it is. Okay. Oh, you're right.
Come on now.
I'm taking mine in Tuesday actually.
You are?
I had to take
I
it
don't.
early.
I know NB.
I don't envy myself.
Okay, here we are at the first.
What is this?
Spirogyra.
It's a group, you know that. That is the same group, Spinal Gyra.
Oh, I was like, a super group? I don't know.
Yeah, that's what I was like.
We're all thinking biology.
That's exactly what I said.
Okay, let me see if I can just go ahead and.
Yeah, I don't even know.
Okay. Let me see if I can focus
That's
it
a
without
cool name
flipping
for a
it.
band.
Oh yeah.
Spider Guy.
Spiral Gyro.
There it
Be
is.
a rock band.
Yeah.
There you go.
Has all spiders.
It's like a little bluish green little spiral.
Football spiral. Yeah.
Now let me see if I can go to the third and get it to focus. Okay. Oh, I passed it. Okay. Spiral gyra, very distinct protist. Ooh. You can see his little baby nucleus, his little red dot. There we go.
Hey, Jamie. How do
Okay.
how do we can tell if it's which domain is it? I don't know. This is these lights back.
These are all protists, so they're going to be in eukarya. Okay? If you notice the first 5 slides, they're going to be significantly smaller because protists are eukaryotes, so they're a lot bigger. So if you look under the first magnification, you can see that's one organism. Okay? Whereas if you pull up D3, trying to find, here we go. Okay, so this is a protist, whereas if you put out a slide of anthrax, it's a giant chunk of little tiny organisms. Okay? Does that make sense? So bacteria are significantly smaller, but they grow in big clusters. 53. My slide box is so sad. Okay, last one I want to show you is not in my slide box, of course.
Spider Guy, which secret group is that in? Does it crawl?
You can't
No, RK Plastidia, I believe.
That's the super
Spiral
group.
dryer. Yeah, let me double check. Yeah, RK Plastidia. Okay. Okay. Lastly, I'm going to show you little amoebas. Not yet. Okay, let me see if I can focus it without. Okay. So there's one. These are amoebas. This is only on the second magnification. See how they're all different shapes? Because all their pseudopodia are in different positions. I'm trying to pick one to focus on so when I zoom in. This is on the third magnification. That's an amoeba.
Bless you.
Oh wow, we're so polite.
You're
Okay.
welcome.
Okay, when it's on the practical, are you gonna give us like a microscope with the slide on it, and then we can just kind of flip through the 3
Yes.
like different
So-
angles or what?
On the practical, you're allowed to touch this because if it's in focus on the highest magnification, it should be pretty close to being in focus on the other. Okay? But I don't want you to move the stage or change the focus. Because if you accidentally get it out of focus, then the person after you is going to have to focus it. That's going to take up more time. And then, so the one that I have you focusing I give you part A and part B to label it, because you only have 3 minutes at each section. So I give you a little extra time, and there's only two parts to that question. So you just leave A blank. Okay. That was all, in my opinion, the hardest part of the practical. All the microscope slides.
Yeah.
For
That's
what?
good.
Fungi
Yeah.
and plant.
This is what you're gonna take from.
I cannot speak for the other sections, but I'm not gonna lie to you either. Since protists are so intense and microscope heavy, I'm not gonna put a microscope slide on fungi or plants.
Oh, you're awesome.
Yes.
We have much, so with protists, you kind of have to see them under the microscope, right? My personal opinion, you can look at a fern and tell me that it's a fern.
Mm-hmm.
You don't need to look at it under the microscope. Okay? So that's my personal opinion. I can't speak for the other lab professors, sorry. Other lab TAs. But I showed you the most important protists and the most important bacteria. I threw in a couple extra just so I wasn't making it too easy.
Still better than a long list because I have no control.
Okay. Last, fungi and plants. Okay. You should be able to label this. Okay? Knowing what order they evolved in will also help you differentiate between the fungi. Okay? Chytrids were first. They were still aquatic, so therefore they have zoospores. Then you have zygomycetes, which were the little furry fungi, which you saw on the strawberry. And then we skipped this one, so I'm not even going to try to say it because I'm going to butcher it. Then you have ascomycetes, which are a little bit bigger. And lastly, you have basidomycetes, which are mushrooms and the biggest fungi. Okay? So learning the phylogeny will aid in your studying later. Okay? Be able to label this.
You said you don't sign off the spelling, right?
That one has a word bank,
Okay.
I believe.
Can you go back 1 second?
Sorry, I'm probably giving y'all way too much information. I just really want you to do really well. If you study, I just want y'all to do well.
I don't think you're giving us
Of course you don't think I'm giving you too much information. Okay, fungi. No, they are heterotrophic. Okay? Again, those are just really cool pictures. On the review, I asked you the 2 different types of fungi. Know the difference. Know the 2 types and know the difference. So saprobes feed specifically on dead matter, and parasites feed on the tissues of other animals or plants. So parasitic fungi, like athlete's foot, feed on our feet. Whereas saprobes, like that one over there, just feed on dead things on the ground. Okay? Oh, sorry. Are you good with the picture? Are you taking a picture or are you just on your phone? Okay. Chitin. Chitin is a support compound. Chitin is in spores. Okay? So the cell walls of fungi are reinforced with chitin, and spores have cell walls. Okay? So there are chitin in spores. Chitin is found in the under part of the mushroom. Okay? I would be very comfortable with the structure of the hyphae, mycelium, and septa. Okay? Hyphae are like the individual little branches. The collection of branches as a whole is the mycelium, and septa are the little cross walls. Okay, so these little lines separating the cells in the hyphae are your septa. Again, you can have complete septa, incomplete septa, which you can see right over there, and then zygomycetes lack septa overall. Okay? Sporocarp and spores are how you distinguish between the different types of fungi. Okay? The sporocarp produces the spores. Okay? I feel like a lot in the next few slides I give you a bunch of words, but if you can remember that the sporocarp produces the spores, it will help a lot. Okay? So the basidocarp produces basidospores. The ascocarp produces ascospores. Zygos, it's not a zygocarp. No. I know, but I don't, is it a zygocarp? I'm not sure, honestly. Okay, chytrids, again, they're the most ancient group. They're at the very top of the phylogenetic tree. They have zoospores. For each group, I expect you to know what type of spores they have and significant, I guess, facts. So no, chytrids have zoospores, and they're the most ancient. Zygomycota, they lack septa. Okay, they have zygospores. What kind of fungi is growing on that strawberry? No, Zygomycota. But specifically, what species, sorry, that was kind of harsh. Didn't mean to say it like that. What species of fungi? You need to know what species of fungi is growing on that strawberry. I told you in lecture, it's on your PowerPoint. That strawberry is real important. Okay? It's on the PowerPoint twice. Okay? It's Rhizopus. Rhizopus is the type of fungi that is. Rhizopus. Rhizopus was also on 2 of the handouts. What's that?
If we put the name, like, isn't it bread fungus?
Yes, it is a bread mold.
If we put that instead of rhizo, whatever, does that work?
I might give you half credit if I'm going to give name. But, okay, so it's a 3-part question. Again, I'm going to ask you what organism, what kind of fungi is that? And then I'll ask you what phyla, so it's in Zygomycota. And then I might ask you another question, like what domain? What domain are we in? Eukarya. Okay. Fungi. What kingdom is this? Fungi. Okay. So even if you can't remember that that's a bread mold or that's Rhizopus, you should get partial credit. Okay. I worded a lot of the identifying questions so that even if you can't get the whole question, I want you to at least get one part of it. Okay.
How'd you say you spell it?
Rhizopus, Rhizopus. Because there are questions on the practical that were on the handouts. Okay. That was on the handout twice. Ascomycota are sac fungi. Asci are the individual little sacs. Ascocarps are the whole fruiting body. And then these are inside of here. This is Connata. Okay? So here's the biggest structure. Inside Connata are ascocarps, which are these little baby structures. And then inside ascocarps are asci. Okay? Basidomycota mushrooms, shelf fungi, puffballs. Basidospores made by the basidocarp. Okay. Those are just cool pictures. From Mario Kart. A Mario fan. This is really cool. I mean, it might be a bonus question. I don't know. If I could wink, I would. Amantia. I think that's all for fungi. Oh, lichens. So if I say true or false, in the taxonomic group lichens, there are 3 types. False. There are 3 types of lichens, but they're not a taxonomic group. Lichens are an example of a symbiotic relationship. That's the purpose of this image. Again, I can't speak for other TAs. The only purpose for this image is to show you that a lichen is the conglomeration of an algae and fungi, or I believe, or cyanobacteria. Most of the time it's algae, but it could be cyanobacteria. Okay? Know the 3 types of lichens. There were examples out last week, so if I give you an example of a lichen, I expect you to say, okay, well that's a foliose lichen. That's crustose. Okay? The only example of fruticose was in that little picture frame thing of the preserved species. We didn't have a live lichen of fruticose. Again, mycorrhizae is not a taxonomic group. It's a symbiotic relationship, mutualism. Okay, plants. Seedless plants have spores. Somebody's phone's ringing.
What'd you just say?
Seedless plants have spores, not seeds.
Thank you.
So true or false, the plants we talked about last week had seeds.
False.
Awesome. Okay. Know that plants exhibit alternation of generations. Okay. I drew the picture up on the board. It's on the PowerPoint. There are a couple questions on alternation of generations and the plant life cycle. Okay. On the review, I made specific questions just over this slide. What portions of the plant life cycle are haploid? What portions of the plant life cycle are diploid? Okay? Do you want me to go over there and draw it again?
Would it be more beneficial to know the one that you drew or that one?
They're the same image. Okay? The way I drew it, I started here. Okay? We still have spores that turn into gametophyte through mitosis. Okay, the gametophyte then turns into gametes through mitosis. The gametes then fuse, or through fertilization or fusion. They're monogamous, or not monogamous, that's not the right word. Synonymous. Sorry. Fertilization or fusion, I will accept either. Okay, they fuse to make the zygote. The zygote, using mitosis, turns into the sporophyte. The only time meiosis is used is to go from a diploid sporophyte to a haploid spore. Okay? Because here you're going from haploid to haploid, haploid to haploid. When you go from haploid to diploid, it's fertilization or fusion. And then you go diploid, diploid. So then diploid to haploid, you have meiosis. Okay?
So like, will we have to identify what percentage is haploid?
No.
Okay.
That's just a mean question. Okay. Know that the haploid is the gametophyte. Diploid is the sporophyte generation. So the sporophyte generation is diploid, so that's why it's orange. The gametophyte generation is blue. So in this instance, this image is better than my image because I didn't draw them in two different colors. Does that make sense? So this is the gametophyte generation, and that's the sporophyte generation. The sporophyte generation is going to be diploid. The gametophyte generation is going to be haploid. Okay. Hepatophyta, know the common name and the scientific name of each phyla. The name at the top of this is the phyla. Okay, if you go back, there are multiple phylas under a clade. Okay, you have liverworts, hornworts, and mosses under the clade Bryophyta. So Hepatophyta, or liverworts, are the phyla, but bryophytes are the clade. Okay, if it helps to say bryophytes and pteridophytes, I'll accept that. Because Bryophyta, in my opinion, should not be a clade name because it makes it confusing. Because there is a phyla called Bryophyta. Personally, I think they should be labeled clade bryophytes, clade pteridophytes.
So you said we need to know the common name and what?
And the phyla. So here, the common name is liverwort, but the specific phyla name is Hepatophyta. Okay? No Hepatophyta is gametophyte dominant. Again, this is all review. It's on your review. What is the thallus? The thallus is the haploid body form of liverworts. What are Jimmy cups? Jimmy cups are on the thallus. Okay? Anthocerophyta. Okay? Anthocerophyta are hornworts. Know that they're gametophyte dominant? I'm not going to ask you what kind of sporophyte. Again, look through the PowerPoints enough so that you can identify the live specimens. So for this portion, I'm going to go ahead and tell you I have 1, 2, 3 live specimens. And you're expected to know what phyla they're in, what species they are, what clade, what domain, what kingdom, depending on the organism is what I ask you. Okay? Bryophyta are mosses. Okay, I have all the mosses still out, just by chance, because I haven't put them up, honestly. So if you did not take pictures of the bryophytes, make sure you do. All of those are bryophytes except Arcanthia. Okay. So when you look at the different species, just because they're all in this bin doesn't mean that they're all mosses. Okay. Arcanthia is not a bryophyte. It is a liverwort, I believe.
Did you say you're gonna post the plant slides?
These are posted.
They're not attached to the OneDrive slide.
Oh, they are.
Me too. I'm looking at it.
It's all in one PowerPoint.
Okay, well then when I downloaded the PowerPoint,
It only downloaded half of it for some reason.
yeah,
Yeah, so it's all in one PowerPoint.
okay.
Vascular tissue evolved after the bryophytes. So if you remember the setup in class, I put the bryophytes over here, then the lycophytes, then the pteridophytes. I did that to show that vascular system evolved from that table to that table. Okay? Lycophytes are club mosses. This is when the vascular tissue evolved. You need to know that.
Can you say that again? I'm sorry.
Lycophyta, that is when they evolved to have vascular tissue. So bryophytes overall. So bryophyta, anthocerophyta, and hepatophyta, none of them had vascular tissues. Yeah. Actually, I think Marcantia is a lycophyte. No, just kidding. Sorry guys, I'm confusing. Marcantia is hepatophyta. Okay, pterophyta is the phyla, pteridophyte is the clade. Okay? Pterophyta are ferns and alleys. Horsetails was fern, true ferns. They have advanced vascular tissue known as siphonostele. That is unpractical. Okay? That their vascular tissue is the siphonostele. Siphonostele is the way the vascular tissue is stood up with the xylem and phloem. The prothallus is part of the pterophyte life cycle. Know that sori are the little dots on the bottom of a fern. Okay? So if I give you a fern, on a practical, this fern doesn't have sori, I don't think. Okay. What phyla is that in? You ready? Pterophyta. Okay. It's a fern, therefore it's in the phyla Pterophyta. Okay. Also, does this fern have sori? There are no little dots on the belly of the leaf. I can't get this to twist the way. There you go. Yeah, there's no dots, so there are no sori. This little dying creature has sori. See the little dots? You don't have to touch them so it doesn't die. Give it some more water. Okay, so we all know what sori are. Okay. I'm pretty sure I've covered every question on the exam. I've at least said it out loud. Yep. Do we have any questions? Anything I need to go back over?
If we wanted to come look at the slides, is there a chance to do that before the exam?
You can either do it now, or you can come during Julia's session, or if you want, there's not any labs tomorrow. Yeah, I would do it today.
What time is Julia's?
I don't know.
Julia's review
Depends on
is
who
tomorrow
comes
at
here
6
tomorrow.
p.m.
Yeah.
I can hear it 6 a.m.
Because I mean, I would have to like go home and then drive back.
So how exactly is it gonna
Do you
be
come to
set?
Anchorage on Monday?
There will
Yeah,
be stations.
but it's like,
And
yeah, I know. I
share
just like,
one
and it's just
question,
like I can go
and
home,
each
take a
question
nap
is
and stuff like that. But I mean,
Library.
yeah.
So we only have
Okay,
that
yeah.
amount of time to answer it, and we can't go back?
Guys, once you're at a station, you are not to return to that station, okay? So this is the setup of your practical.